The specific goals of this experiment are to non-invasively assess skeletal muscle oxidative capacity, to assess exercise skeletal muscle blood flow regulation, and to measure reactive hyperemia.
These methods can help answer important questions related to the pathophysiology of exercise intolerance and cardiovascular disease.
The main advantage of this technique is that we can non-invasively assess macro and microvascular function and skeletal oxidative capacity in any clinical or laboratory setting.
Generally, individuals new to this method will struggle because of the operator-dependent nature of near-infrared spectroscopy. It is important to achieve consistent and accurate placement of the probe, as small deviations from optimal placement could have a large impact on signal quality.
Begin with instructing the subject to lie supine with their legs inside an LBNP chamber so that their belt line is approximately even with the opening of the LBNP box. Next, optimally place 3 electrocardiogram electrodes on the subject, 2 in an inferior midclavicular location, and 1 on the subject's left side medial to the iliac crest. To proceed, place a non-invasive blood pressure monitor module on the subject's dominant wrist. Make use of properly calibrated finger blood pressure cuffs. Place them on each finger. Now instruct the subject to grasp an HGD with their non-dominant hand in a slightly abdominal